Hello students, welcome to EPG Pathshala. I, Professor Ramesh Kothari, Professor of Microbiology, UGC CS Department of Biosciences, Saurashtra University Rajkot, Gujarat. Today we are going to discuss about citric acid cycle from the paper Metabolism of Carbohydrate. Let me see in introduction. The TCA cycle, also known as citric acid cycle or Krebs cycle. It is a cyclic series of enzymatically catalyzed reactions carried out by a multi-enzyme system. It oxidizes the acetyl group of acetyl coenzymes A to carbon dioxide, reduce nicotinamide adenine dinucleotide, that is NADH, hydrogen, and reduce flavin adenine dinucleotide, that is FADH2, using oxidized nicotinamide adenine dinucleotide, that is NAD+, and oxidized flavin adenine dinucleotide, that is FAD, as electron acceptors. The reactions of the TCA cycle occurs within the inner mitochondrial membrane. They are thus sequestered from the reactions of glycolysis and fatty acid synthesis, which are cytosolic systems but are in competition with oxidation of fatty acids. Electrons from the TCA cycle and oxidation of fatty acids are transferred to acceptors in electron transport chain, which is also located within the inner mitochondrial membrane, as are the enzymes of oxidative phosphorylation. Now let's see the objective of this lecture. In this lecture, we will discuss history and introduction of citric acid cycle, conversion of pyruvate to activated acetate. Then we will discuss various reactions of citric acid cycle and the amphibolic nature of citric acid cycle. As we all know that Hans Krebs, a German-born British biochemist who received a Nobel Prize in Physiology for the discovery of the Krebs cycle. Krebs discovered when pyruvate and oxaloacetate combined, the citrate is formed, thus explaining the reason why the reaction form a cycle. Krebs also discovered the urea cycle with another biochemist, that is Kurt Hanslitt. In this cycle, ammonia is converted to urea in the tissue of mammals. Being far less toxic than ammonia, urea is excreted in the urine of most mammals. By 1930, it was established that the addition of lactate, acetate, succinate, malate, alpha-ketogluteric acid, that is carboxylic acids, and citrate as well as isocitrate, when added to muscles, means that they stimulate oxygen consumption and release of carbon dioxide. In 1935, Albert Sand and Georgi explained the conversion of succinate to fumarate, fumarate to malate, and malate to oxaloacetate. Karl Marius and Franz Knupp explained the conversion of citrate to cis-aconitate, cis-aconitate to isocitrate, and isocitrate to alpha-ketoglutarate, and alpha-ketoglutarate to succinate, succinate to fumarate, fumarate to malate, and malate to oxaloacetate. Marcius and Knupp showed that pyruvate and oxaloacetate could form citrate non-enzymatically by the addition of peroxide under basic conditions. Krebs showed that succinate is formed from fumarate, malate, or oxaloacetate. This is interesting since it was shown that the other way worked as well. As shown in the reaction, pyruvate can form citrate enzymatically. The interconversion rate of the intermediate was fast enough to support the respiration rate. The citric acid cycle is also called the Krebs cycle or the carboxylic acid cycle, and it is the hub of metabolic system. It accounts for the majority of carbohydrate, fatty acid, and amino acid oxidation. It also accounts for a majority of generation of this compound and other as well. In eukaryotes, the TCA cycle occurs in the matrix of the mitochondria, where in prokaryotic cell, the TCA cycle occurs in the cytosol. Figure shows the reaction of TCA cycle. All cyclic pathways have to have an acceptor molecule. Here, oxaloacetic acid for the input and acetyl part that comes from pyruvate. If you see in this figure, 2 carbon moiety of acetyl-CoA when combined with 4 carbon moiety oxaloacetate, it forms citrate, and through a series of reactions, the citrate convert to isocitrate, then alpha-ketoglutarate, then succinyl coenzyme A, succinate, fumarate, malate, and again oxaloacetate. This oxaloacetate again combined with acetyl-CoA and cycle continue. You can see that at certain stage, certain reactions are decarboxylation reactions, some are dehydrogenation reactions, and it generates ATP through electron transport chain. Certain reactions are hydration reactions, and all are regulated by a certain enzymes or a set of enzymes. Now we will discuss several enzymes and enzyme system involved in tricarboxylic acid cycle. To start with, pyruvate dehydrogenase complex. It is a multi-enzymes complex containing 3 enzymes associated together non-covalently. Enzyme 1, pyruvate dehydrogenase. It uses thiamine pyrophosphate as cofactor bound to enzyme 1. Enzyme 2, that is dihydrolipoyl transacetylase, lipoic acid bound coenzyme as substrate, And enzyme 3, that is dihydrolipoyl dehydrogenase, FAD bound NAD plus as substrate. There are obvious advantages of multi-enzyme complex, and these advantages are higher rate of reaction because product of one enzyme act as a substrate of other and is available for the active site of next enzyme without much diffusion. Second advantage is minimum site reaction, and third is coordinated control. Here coordinated control is very important as the concentration of substrate and the concentration of product should be maintained throughout the reactions. The above figure shows a transmission electron micrograph of localization of pyruvate dehydrogenase complex. You can see in the figure several part of enzymes are indicated in different color. A red color shows the part of enzyme 1, green color shows enzyme 2, and yellowish color shows enzyme 1. Now, reaction of a pyruvate dehydrogenase complex. Figure shows a reaction of conversion of pyruvate to acetyl coenzyme A catalyzed by a pyruvate dehydrogenase complex. This reaction is exergonic and produces 33.4 kilojoule per mole energy. In this case, the pyruvate is converted to acetyl CoA with release of carbon dioxide. NAD plus is converted to NADH, and pyruvate dehydrogenase complex continuously converting pyruvate to acetyl CoA till the pyruvate becomes available. This figure shows various sites of reaction located in pyruvate dehydrogenase complex. If you see the site 1 where pyruvate undergo decarboxylation and hydroxyethyl is produced, then by the action of acyl lipolysine, the oxidized lipolysine and the acetyl CoA takes coenzyme A. Then later on this NADH is converted, NADH when react with hydrogen it converted into NAD plus. So you can say that enzyme 1 pyruvate dehydrogenase, enzyme 2 dihydrolipoids transacetylase, and enzyme 3 dihydrolipoyl dehydrogenase work in coordination to convert pyruvate into acetyl CoA. Now let's have a look of several reactions catalyzed by a specific enzyme in TCA cycle. Step 1. Acetyl and oxaloacetate react to generate citrate. Here coenzyme ASH gets free and reaction is catalyzed by enzyme citrate synthase. During step 2, citrate rearrange to isocitrate and reaction is catalyzed by enzyme acotinase. In step 3, it is an oxidative decarboxylation reaction. NAD plus convert into NADH. Removal of carbon as carbon dioxide take place. Isocitrate converted into alpha ketoglutarate, and reaction is catalyzed by enzyme isocitrate dehydrogenase. During step 4, this reaction converts alpha ketoglutarate into succinyl-CoA. Oxidative decarboxylation occurs again to remove carbon, and reaction is catalyzed by enzyme alpha ketoglutarate dehydrogenase. In step 5, during this reaction, ATP forms via substrate level phosphorylation. Succinyl coenzyme A converted into succinate, and the reaction is catalyzed by enzyme succinate thiokinase, which is also known as succinyl coenzyme synthetase. Now, during reaction 6, step 6, during this reaction, succinate oxidized to fumarate, FADH2 produced, and the reaction is catalyzed by enzyme succinate dehydrogenase. During step 7, during this reaction, fumarate hydrated to produce malate. The reaction is again catalyzed by enzyme fumarase. In step 8, during this reaction, the malate is oxidized into oxaloacetate, and the reaction is catalyzed by enzyme malate dehydrogenase. The TCA cycle is an amphibolic pathway that is involved in both anabolic as well as catabolic process. Anabolically, the TCA cycle provide route for a synthesis of biosynthetic precursors, and catabolically it provide the degradation of 2-carbon acetyl residue, which is derived from carbohydrate, fatty acids and amino acids. The TCA cycle is an amphibolic pathway that is involved in both anabolic as well as catabolic processes. Anabolically, the TCA cycle provides route for synthesis of biosynthetic precursors, and catabolically it provide the degradation of 2-carbon acetyl residue, which is derived from carbohydrate, fatty acid and amino acids. Citrate synthase, that is a formation of citral coenzyme intermediate. Binding of oxaloacetate to the enzyme result in a conformational change which facilitate the binding of next substrate, the acetyl coenzyme A. There is a further conformational change which lead to formation of product. This mechanism of reaction is referred as induced fit model, and this reaction generate, again this is an exergonic reaction, and it generate 32.2 kilojoule per mole energy. Here the enzyme catalyzes the isomerization reaction by removing and then adding of water to cis-aconitate at different position. Isocitrate is consumed rapidly by the next step, thus deriving the reaction in forward direction. So first citrate is converted to cis-aconitate by the action of aconitate aconitase enzymes, and this reaction is dehydration reaction. And cis aconitate by the action of aconitase, again addition of water molecule results in the formation of isocitrate, and now isocitrate is ready to enter to further reactions. The real work begins in the third step of the TCA cycle. Isocitrate dehydrogenase remove one of the carbon atom from isocitrate, forming carbon dioxide, and transfer electron to NADH. There are two isomers of isocitrate dehydrogenase enzyme. One uses NAD plus and other uses NADP plus as electron acceptor. The fourth step is performed by a huge complex composed of three types of enzymes connected by a flexible linkers. A lot of things happen in this complex. One more carbon atom is released as carbon dioxide. Electrons are transferred to NADH, and the remaining part of the molecule is connected to coenzyme A. Alpha ketoglutarate dehydrogenase is a complex of different enzymatic activities similar to the pyruvate dehydrogenase complex. It has the same mechanism of reaction with enzyme 1, enzyme 2 and enzyme 3 units. NAD plus is an electron acceptor in this case. This is the only step in the TCA cycle where ATP is made directly. The bond between succinate and coenzyme A is particularly unstable and provide the energy needed to build a molecule of ATP. In mitochondria, the enzyme actually creates GTP in the reaction, which is converted to ATP by the enzyme nucleoside diphosphate kinase. Succinyl coenzyme A synthetase. The succinyl coenzyme A, like acetyl coenzyme A, has a thioester bond with very negative free energy of hydrolysis. In this reaction, the hydrolysis of this thioester bond leads to the formation of phosphoester bond with inorganic phosphate. This phosphate is transferred to histidine residue of the enzymes, and this high energy unstable phosphate is finally transferred to GDP, resulting in the generation of GTP. In the sixth step, succinate dehydrogenase found in the membrane links its citric acid cycle task directly to the electron transport chain. It extracts hydrogen atom from succinate and transfer them first to carrier FAD and finally to the mobile electron carrier ubiquinone. Enzyme succinate dehydrogenase carry out oxidation of succinate to fumarate. This is the only citric acid cycle enzyme that is tightly bound to the inner mitochondrial membrane. It is an FAD-dependent enzyme. Malonate has a similar structure to succinate and is competitively inhibit SDS. The seventh step in tricarboxylic acid cycle is catalyzed by enzyme fumarase, which add a water molecule to get everything ready for the final step
of the cycle. Enzyme fumarase carry out hydration of fumarate, converting it into a malate. It is a highly stereospecific enzyme. Cis-malate, the cis form of fumarate, is not recognized by these enzymes. The enzyme is exergonic and generate 3.8 kilojoule per mole energy. So if we summarize the reactions of TCA cycle in nutshell, the 2-carbon acetyl group regenerated in pyruvate dehydrogenase complex reaction enters the TCA cycle, and the 2 molecules of carbon dioxide are released in cycle. Thus, there is complete oxidation of a 2-carbon during one cycle. Although the 2-carbon which enters the cycle become the part of oxaloacetate and are released as carbon dioxide only in the 3rd round of the cycle. The energy release due to this oxidation is conserved in the reduction of 3 molecules of NAD+, 1 molecules of FD, and synthesis of 1 GTP molecule, which is converted to ATP. You can see these different events in this figure. The oxidation of 1 glucose yield 2840 kilojoule per mole energy. Energy obtained by biological engine, if you count, it is 32 ATP multiplied by 30.5 kilojoule per mole. It is equivalent to 976 kilojoule per mole. Thus, 34% efficiency is obtained if calculations are done using standard conditions. But if concentration in the cellular conditions are taken into account, the efficiency is close to 65%. This table shows stoichiometry of coenzyme reduction and ATP formation in the aerobic oxidation of glucose via glycolysis, the pyruvate dehydrogenase complex reaction, the citric acid cycle, and oxidative phosphorylation. The figure shows the amphibolic nature of citric acid cycle. This pathway is utilized for the both catabolic reactions to generate energy as well as for anabolic reactions to generate metabolic intermediate for biosynthesis. If the carboxylic acid cycle intermediates are used for synthetic reactions, they are replenished by anaplerotic reactions in the cell, which is indicated by red color in the figure. Again, this slide figure shows the amphibolic nature of citric acid cycle. You can see the glyoxysome environment and mitochondrial matrix. Here, if you see in glyoxysome, the acetyl-CoA, which is synthesized from fatty acid, enters in the cycle and produce citrate when it combined with oxaloacetate, which results in the formation of isocitrate, which in turn converted into glyoxylate, releasing the succinate. Again, by the addition of acetyl coenzyme, it converted into malate, and thus the glyoxylate cycle gets complete. If you see in the mitochondrial membrane, the succinate which is produced in the glyoxysome enters in the citric acid cycle, and it converted into fumarate. Then fumarate converted into malate and oxaloacetate, and again citrate. Thus, the citric acid cycle also simultaneously operates. Here, the malate, when release from the mitochondrial matrix, converted to oxaloacetate, and by the process of gluconeogenesis, it produces hexoses and sucrose. This figure also shows the amphibolic nature of citric acid cycle. Here acetyl CoA when convert into isocitrate, and isocitrate takes two route. Isocitrate is converted into succinate glyoxylate and then oxaloacetate to glucose to amino acid and nucleotide synthesis. In another route, isocitrate converted into alpha ketoglutarate through TCA cycle, and by the process of oxidative phosphorylation it generates ATP. The figure shows the points of regulation of TCA cycle, the rate of controlling enzymes, that is citrate synthetase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. The regulation of the activity of this enzyme is achieved by the substrate availability, product inhibition, and allosteric inhibition of activation by other intermediate. If you see in figure, you can see this red color cross point. These are the points where the major regulation of TCA cycle take place.
So, student, we have discussed about various reactions of TCA cycle. Now, if we conclude this TCA cycle, we know that when glucose enters in the TCA cycle, before it enters in the TCA cycle, it passes through the glycolytic pathways. We know that there are two phases of glycolytic pathways. One is preparatory stage and second is ATP synthetic stage. In first stage, ATP are consumed. In second stage, ATP are produced. And then glucose is converted into acetyl CoA. Now, under aerobic condition, by the various enzyme system, acetyl CoA is pyruvate is converted into acetyl CoA. Now, acetyl coenzyme A is enters in the TCA cycle, where it passes through various reversible and irreversible reactions, and it produces again energy at different level. Certain ATP are produced at phosphorylation and certain are produced through electron transport chain, and thus it provide plenty amount of energy which is required for the metabolism of cell. So, we can say that TCA cycle provide a complete route of aerobic catabolism of glucose, generating sufficient amount of energy required for cell metabolism as and when required. It is a central metabolic pathways indispensable for cell metabolism. We know that TCA cycle also provide several intermediate metabolite which act as a substrate for the synthesis of other various metabolites. For example, it is also useful for the synthesis of amino acids. Thus, the TCA cycle, it is a central metabolic pathways and very important for existence of cell, and it metabolize under aerobic condition various carbon structures. So, this is all about the citric acid cycle. Thank you.